Now I'd like to introduce our second presentation for the day, Yokogawa Fluid Imaging Technologies. Thank you for being one of the sponsors of today's webinar. Now I'd like to introduce you to the speakers for this session. Savannah Stresser. Savannah is the Aquatics Business Development Manager for Yokogawa Fluid Imaging Technologies. She works closely with FlowCam users to identify and optimize applications where FlowCam adds value, including shellfish hatcheries and research programs. She has 10 years of experience in environmental science, including business and nonprofit organizations dedicated to water quality, monitoring, and aquaculture education. She's based in Southern Maine. Next, I'd like to introduce Leah Ann Gabala Smith. Leah is the Marine Aquatics Sales Manager for Yokogawa Fluid Imaging Technologies. She works collaboratively with research hatcheries and monitoring programs to evaluate the efficiency high-speed plankton imaging could bring to their workflows. She has over 2 decades of specialized experience in aquatic sciences and phytoplankton taxonomy, with a particular focus on harmful algal bloom species in shellfish growing areas. She's based in southeastern Virginia. Without further ado, I'll pass it over to Savannah and Leah for their presentation.
Great. Thank you so much, Alex. We're really excited to be here and participating in Aquaculture Technology Webinar Week for the first time. So we're today going to be talking about how flow imaging microscopy is being utilized in a variety of shellfish hatcheries and research programs to help expand capacity for counting and measuring organisms inside the hatchery, like eggs, larvae, and microalgae, as well as external threats from harmful algal blooms. So anytime that we've been approached by a hatchery or a research team in the past, we often hear some version of the following challenges. So for example, when it comes to counting microalgae that are used to feed shellfish larvae, hatchery managers are often frustrated by how difficult it is to get consistent counts from person to person and how much time that takes out of every single day. Now, the same is true when we talk about oyster larvae as well. Precision is particularly challenging, the smaller the larvae are. People often joke that larval rearing is just as much of an art as a science, but all of the hatcheries that we work with are really trying to bring as much of a data-driven approach to their operations as possible. And the one that really breaks my heart the most, though, I think, is, when people say that the biggest stressor they have is having to tell their boss that they have way fewer seed than they thought they would, and they never have a really clear reason why. So not having as many larvae as you predicted, is really, really challenging, both from a scientific perspective as well as from a business perspective. So more and more hatcheries are looking for more automated techniques to help solve these types of challenges. And flow imaging microscopy is one such technique that checks a lot of these boxes. So this technique combines the optical capabilities of microscopy with digital imaging to help reduce inter-operator variability, but also performing this imaging in flow, which means we can do it faster than would otherwise be possible. So to give you a benchmark, it takes about 5 minutes to run 1 milliliter of sample, targeting organisms up to about 100 microns, which are some of the trickiest ones to count on the microscope. And the standard deviation between replicates could be as low as 1%, depending on the sample. And on top of that, you also get pictures and measurements that you can refer back to at any time. So this is a quick visual of what data, acquisition looks like. My video isn't playing. There we go. We'll try it again. This is a quick video of what data acquisition looks like if you were sitting down at the FlowCam watching a sample pass through. On the left, we have a live feed of what's passing in front of the camera. And every particle that passes in front of the camera that meets my acceptance criteria automatically gets cropped, counted, measured, and saved to a database where it can then be filtered or classified depending on the questions that I'm trying to answer. So some of the applications that we're going to talk about for flow imaging microscopy in shellfish hatcheries today include research projects focused on reproduction and fecundity, larval culture, as well as harmful algal bloom monitoring. So we're going to take a quick virtual tour across the east coast of the US, visiting a few different shellfish hatcheries. Now we're going to start up north in Maine with the Down East Institute. This is the easternmost marine research lab in the US. It's the marine science branch of the University of Maine at Machias. And their mission is really focused on wild and cultured fisheries with an emphasis on research, education, and innovation. So Brian Beal and Tessa Houston are a couple of the FlowCam users at the Down East Institute, and they do really amazing work with softshell clams. For those of you who don't know, commercial landings of softshell clams have been declining here in Maine for some time, and there's a lot of interest from the industry to see how we might be able to turn this trend around. One of the questions that Brian and Tessa have set out to answer is whether larger clams actually make more baby clams than smaller ones. And if they do, this would make a really strong case for limiting harvest of the largest clams because they produce the most eggs and are most important to the recovery of the fishery. A really great analogy is lobster. A lot of people who aren't from Maine may not appreciate the fact that not only is there a lower limit on the size of lobsters that can be kept for sale, there's also an upper size limit. So whether a lobster is too small or too large, that lobster has to be thrown back into the ocean because those largest lobsters produce the most eggs, and again, are most important to the sustainability of the fishery. But until now, we really didn't have any data of this kind to help explain whether or not the same was true for softshell clams. So that's something that Tessa and Brian have been able to do using the FlowCam. So they used FlowCam to count and measure the eggs that female clams of different shell sizes produced at a variety of sites across Maine. So the team found that indeed bigger females did generally produce exponentially more eggs than smaller ones. So based on data like this, legislation is actually being proposed that's going to help the fishery recover by conserving the number of large clams, adult clams that can be harvested. These are some examples of what those clam eggs actually look like on the FlowCam. You can see up here in the top row, these eggs actually also show, based on the images that we have, the polar bodies that form when those eggs are fertilized. So you can see not only how many eggs are being produced, but especially when they're spawning, you can also take a look at which are the proportion of eggs that have been successfully fertilized. And one of the things that we're really interested in continuing to explore with our partners at the Down East Institute in the future is how those numbers compare, and also looking at developmental abnormalities in eggs as well. Down East Institute has also used the FlowCam to check for abnormalities in their microalgae cultures, so looking for contaminants. This is above and beyond just being able to count the algae and speed up some of those regular operations in the hatchery. Last but not least, being the Marine Research Lab of UMaine Machias, the Down East Institute also uses FlowCam to assist with training new students and technicians and for conducting outreach. Next, we'll move a little bit further south to the NOAA Milford Lab in Connecticut. This is where they're using FlowCam as part of an East Coast oyster breeding program to help cross lots of different families of oysters to find the ones that are optimized for changing conditions, whether that's temperature or salinity, so that ultimately we end up with oysters that are really designed to thrive in different regions up and down the East Coast. So they use the FlowCam for a wide variety of applications. In addition to counting their larvae, they also use it, like we mentioned with the Down East Institute, to detect abnormalities. So in this example, we can see a few different eggs that are really nicely well formed, like in the examples we saw from the Down East Institute, but we can also see many examples of eggs that are not doing so well. And this is really important for breeding programs like at the Milford Lab, because we know that the sooner that they know they have a problem with a particular cohort, the faster they can pivot. Next, they also use the FlowCam to check the viability of the oysters. So what's the proportion of oyster larvae that are live or dead? And they do this with some basic value filters, looking at the intensity of the larvae on the FlowCam, as well as doing some experiments with neutral red, which is a vital stain. Last but not least, they've also built libraries of 5 different commercial species, so not just oysters, but also mussels, as in this example, and scallops, looking at how they progress from day 1 all the way through their final set. And now I'll turn it over to Leah, who's going to talk a little bit about some of the external threats and how FlowCam helps with HAB monitoring programs related to shellfish.
Thank you so much, Savannah. Another essential component of hatchery and growing area safety includes monitoring for harmful algal blooms as part of a federal and state cooperative effort to ensure the safe growth and harvest of shellfish by avoiding the HAB species that produce biotoxins. We will continue our southbound journey through southeastern Virginia and to coastal Georgia, where shellfish researchers work to better understand these organisms and how they might impact shellfish operations. On the shore of the York River in southeastern Virginia, the Virginia Institute of Marine Science is home to the Batten School of Coastal and Marine Science and the Acuff Center for Aquaculture. Researchers here study seasonal blooms with the potential to impact hatchery and growing operations. Their work is used to make operational decisions in their own research hatcheries and to train future aquaculturalists. As a state agency tasked with providing support to regional growers, harvesters, and regulatory agencies, their monitoring program has real-world applications. Kim Reese, a molecular geneticist specializing in shellfish pathogens and toxin-producing HAB species, has incorporated imaging technologies into her research and the monitoring efforts they conduct for the Virginia Department of Health. Virginia marine and estuarine waters have a diverse group of HAB species which can impact shellfish harvests and hatchery operations if they enter the hatcheries with intake water. Screening for these targeted HABs is essential to maintain the health of hatchery-grown shellfish and monitor the safety of wild harvested shellfish. Savannah Mapes, pictured here, is a PhD candidate in Kim's lab who has been working with imaging technologies at VIMS. Taxonomic enumerations on environmental samples collected along a salinity gradient on the York River were analyzed microscopically. Samples containing targeted HAB taxa were run on FlowCam to build filters. Image libraries of species were built for different orientations to allow for better filter performance. To test a filter's performance for screening environmental samples for a target HAB, a sample was collected during a large heterocapsa bloom in April of 2024 and run on FlowCam. The heterocapsa filter recognized the target during initial validation testing and provided reliable cell counts when compared to those obtained on the microscope. In Kim's words, FlowCam is transformative when conducting cell counts. In addition to research and monitoring, Kim Reese and her fellow researchers at VIMS also train future aquaculturalists in growing and hatchery operations and conduct outreach activities with local industry partners. This includes HAB taxonomy training and teaching folks to recognize potentially harmful algal species. Using the digital libraries produced by FlowCam, access to resources online will contribute to building a regional toolbox to help in this effort. Here you can see a few mosaics of some shellfish HABs commonly found in these waters. Last but certainly not least, we'll take one more jump down the coast, this time from Virginia to Georgia. So far, we've talked about using FlowCam to count and measure eggs and larvae and assess their chances of survival. But it can also be used to monitor for harmful algae that could affect shellfish in the hatchery. This is Mallory Mintz from Skidway Institute of Oceanography, who uses FlowCam to monitor HABs. She shares her data with the University of Georgia Oyster Hatchery next door. Skidway started using FlowCam to ramp up HAB monitoring after a mysterious crash happened in the late 2010s that caused a mortality event at the hatchery. They found that the crash coincided with a high incidence of Akashiwo. Moving to a more rapid screening tool like FlowCam has allowed Skidway to sample more frequently and get results out to the hatchery much faster than ever before. It's also enabled Mallory to increase the spatial resolution of her sampling efforts to better understand Akashiwo dynamics at multiple depths and at multiple sites. A lot remains to be uncovered about the mechanism that links Akashiwo to oyster mortality, which is one of the many things Mallory plans to investigate further as she embarks on her PhD. Whether it's working with eggs, larvae, or harmful algae, we think this quote resonates with a lot of people, and it's one of our favorites here at FlowCam. First of all, the Haskin Shellfish Lab named their FlowCam Odin, which is an immediate gold star, hands down one of our favorite FlowCam names out there. But what we love most about what Daphne says here is how important Odin is to the work they do, whether it's higher throughput, increased precision, or gaining more automated measurements. Odin and other FlowCams like him are workhorses that are helping shellfish research labs accelerate and expand the work they are doing. And a large commercial hatcheries are starting to catch on to this too. There's no perfect tool out there, and there are still things we are going to continue investigating and improving upon. And we're very grateful to get to work with so many wonderful users. Well, thank you all so much for attending our discussion today.
Thank you guys so much for that awesome presentation. Now let's take some questions from the audience. The first one that I'm seeing is, how fast can you run a sample on FlowCam?
You can hear me pretty good?
Yep, you're coming through loud and clear.
Okay. Well, for your typical sample that you would be running, about 1 milliliter of sample for organisms smaller than 100 microns, it would take approximately 5 minutes to run that sample and get your counts. These would be for phytoplankton, shellfish eggs, and some of the younger shellfish larvae up to about 5 days. When you needed to increase your size, your flow cell then would allow you to capture older larvae or the larger organisms. You could run the same volume of sample in about half that time because your flow cell's larger.
Awesome. Thank you so much. Got another one here asking, what consumables does FlowCam require?
Well, on a day-to-day basis, for your regular just running samples, you would be using the way you load your instrument is you place a disposable pipette tip. Any type that you have in your lab it works fine. And those disposable pipette tips you would change out in between each sample to reduce the potential for cross, like washing out something from one sample to the next sample. Over the course of a year or two, you would be using some of our focal beads or maybe some Citrojet cleaner to give some of the components a good wash. But you would not need to replace that every year. You might buy a bottle every, depending on how much you're using it, maybe every 2 or 3 years or something like that.
Awesome. Thank you. Got another one here asking, does it ever clog? If so, how do you prevent or mitigate that?
Well, I'll answer that in 2 parts, but it is easy to avoid clogging, and then also easy to fix a clog yourself in-house. So we provide you some best practices and SOP for determining what filter to run your sample with prior to deploying it into the instrument. And then also we provide a little chart as to what size flow cell goes best with your sample. And then if you do get a clog, you can usually flush the clog through just with the flow cell right in the instrument. You just kind of go to your fluidics page in the software and you give it kind of a high speed flush forward. You can even run the instrument backwards. And for a really stubborn clog, which might happen with some of your bigger shellfish larvae, we do give a syringe to you at the time of purchase as part of your setup kit. And you can flush really forcefully once you remove the flow cell out. And I've rarely had to use that method, but typically using that method will clear your clog. And all that can be done in-house, like by yourself, by your technician.
Perfect. Awesome. Thank you. Got a bit of a multi-question here, so bear with me while I get through it. So this
Okay.
asking a couple of questions related to shellfish mortality events related to phytoplankton. What is the
Okay.
practical lower size limit for phytoplankton identification, and how much AI training is required for phytoplankton identification prior to analysis?
Okay, so you cut out just a little bit. You were asking me what size are the phytoplankton? Is that what you were asking me?
Yeah, what is the practical lower size limit for phytoplankton identification?
Okay, so the instrument can take images of particles between 2 microns and a millimeter. Uh, the smaller particle, the smaller size range of the particle, like if you were doing an environmental sample, you would be detecting particles as small as 2 microns, 3 microns, 4 microns, but you might not be able to tell the difference between things at 2 or 3 microns. But once you get up to, 8 microns, 10 microns, you can start identifying them. So the very, very lowest size of that range, especially at the 10x, you might not be able to tell the difference for those few. But if you think about what you do on a microscope for an environmental sample, that's the same case on a microscope. So you're not going to be adding, you're not going to be decreasing accuracy in that regard. And then in terms of AI, the software that comes as part of FlowCam is called Visual Spreadsheet, and that uses statistical measurements. Each particle that is captured, an image is captured, over 40 different measurements are taken, lengths, or edge gradient, color, and another, a whole suite of measurements. And so when you're creating filters and libraries and things of that nature, when it does the sorting within Visual Spreadsheet, it's using a statistical measurement. It's not doing AI, but there are different tools out there that you can export this data, just like you would do with some of the other images imaging instruments that are out there. You can export the data in a very usable format and put that into some of those tools like Ecotexa.
Perfect. Awesome. Thanks so much, Leah. Got another one here asking, can the FlowCam system be automated to sample, for example, every hour?
Did you say every hour?
Yep.
Okay, so this is actually a benchtop instrument, and so it's not deployable, let's say on a platform, but many users do bring the instrument shipboard and are able to process samples while underway, while cruising. But you do load the sample yourself into the instrument. There is also the ability to do large quantity of sample, where you would run a pump and you could like pump that through to have continual sampling. But again, that would be within a laboratory environment of that nature.
Perfect. Awesome. Got another one here asking, could this be used for plankton counts on sea sites?
On sea sites, so if what I think you're asking me is if it can run samples on marine water and freshwater, coastal waters, and that answer is yes. You can also use this instrument to run samples of preserved material as well as live material. And we have a whole list. And if you use an unusual preservative, we're happy to check to make sure it is compatible with our components. But it's fine to use Lugol's preserved material, glutaraldehyde preserved material, etc. And marine samples versus freshwater. Marine samples, you do want to make sure that you are cleaning the components really well, like you would anyway with freshwater. But you always, once you get salt involved, you do want to make sure you rinse really nicely so that flow cell stays nice and clean.
Perfect. Awesome. Just a couple more questions here. This one's asking, how much space does FlowCam take up?
It's not as big as you might think it is. Our engineers have made it to the point where I can lift it up and down from a travel box onto anybody's counter. And if I could do it, I'm sure you guys could do it. It's not It's not very big. I would say it's about, I wrote down the size in case somebody asked me the question, but I can't find where I wrote that. But it's about, it's not even as big as a medium microwave, let's say.
Perfect. Awesome. Looks like we just have one more question here, and this one's asking, what does the training process typically look like?
Well, so the training process is typically at the time of installation. So every instrument is built to order because you have, a variety of configurations to choose from, trigger mode, color, etc. And then once the instrument is delivered, our field engineer comes to your site and unboxes it, and he performs a QA on the instrument to make sure it's performing properly after shipping. And at that time of installation, he usually will stay for 1 or 2 days and train your whole staff. And if you need additional training or you have staff turnover, we're always very happy to tag that on to our regular annual maintenance trip. When they come to your facility, you can always add on an additional day of training. And then we also have, with our different service plans, you have access to what's called FlowCam University. And that's an online resource that you can access that has tech notes and videos. And you always have access to myself to put you in touch with the right person in maybe our applications lab if you're struggling with something in the software. And then our customer service team is always available to help you walk through any issues that you might have.
Perfect. Awesome. Thanks so much, Leah. Really appreciate you taking the time to be here today and answer all those questions.